All right, we are at the last chapter 49.4 lecture, and I promise this will be a short one. I tend to go off on tangents. I get excited about the nervous system, but this will be a short lecture, don't worry. Okay, so I finally just wanted to talk very briefly about learning and memory. How do we learn? So learning is brought about by changing connections between neurons, okay? So it's the result of forming new synapses in the brain and also strengthening existing synapses. So this is not the result of creating new neurons. As I mentioned before, your central nervous system has a very limited ability to make new cells after birth. You're born with the vast majority of the brain cells that you're ever going to have in your entire life. So the way that we learn things and form new memories is by strengthening the connections that exist between neurons. So this creation of new synapses or the strengthening of existing synapses is referred to as neuronal plasticity. So the word plasticity refers to changes, and then neuronal refers to neurons. So oftentimes neuroscientists like to use the phrase, the neurons that fire together wire together. So we'll talk in this lecture a little bit about the mechanism by which if two neurons frequently communicate with each other, they can actually enhance their communication. And this is going to be the cellular basis of learning and memory. All right, to step back and talk just a little bit about the theory of memory. So there are two different types of memory. There's what's called your short-term memory and your long-term memory. So your short-term memory is sort of ideas that you hold in your head. Like if I told you a phone number, you would hold that in your short-term memory. If I told you to repeat back some digits. But then eventually, unless you were using that phone number for something important, it probably wouldn't make it into your long-term memory. So this is things that you remember that happened days or weeks ago or years ago. Both of them are going to involve the storage of information in the cerebral cortex. They're also going to involve the involvement of other brain areas, including what's called the hippocampus, which we mentioned as being part of the limbic system. The hippocampus seems to be particularly important for the conversion of these short-term memories into long-term memories. This process is called consolidation. And again, it's not a coincidence that the hippocampus is part of the limbic system, which is involved in our connection with emotions. Because again, the more of an emotional connection you have with something, the greater the chance that that short-term memory is going to be converted into a long-term memory. I wanted to also mention that another thing that's important for consolidation of memory is sleep. So if you remember very early on in Bio 151, I talked to you about the fact that it's been shown that something called distributed practice has been shown to enhance retention of study material. So distributed practice means that you study something over a longer period of time. So you study biology every day for 30 minutes rather than waiting until the end of the week and studying it for 3.5 hours. It's the same amount of time, but you get greater retention if you study it every day for 30 minutes versus one day for 3.5 hours. And that seems to be at least partly because of the role of sleep in consolidating those short-term memories and making them into long-term memories. Okay, and then finally, I wanted to talk a little bit more about the mechanism by which two cells can strengthen their synaptic connection, and a cell can increase its sensitivity to signals from another cell. This process is called long-term potentiation, or LTP. So this is showing you a picture of what this synapse looks like prior to LTP happening, right? So here we have the presynaptic neuron, and it communicates with this postsynaptic neuron. Okay, so the presynaptic neuron, like many central nervous system neurons, uses the neurotransmitter glutamate, which is going to have an excitatory effect, hopefully you remember from last week, on the postsynaptic neuron. And you can see here that glutamate works by binding to a type of receptor called an NMDA receptor. And the NMDA receptor is going to allow magnesium ions to enter the cell. So glutamate binds to the NMDA receptors. This is going to allow also sodium and calcium to enter cells, and that's going to cause excitatory postsynaptic potentials. Again, glutamate is an excitatory neurotransmitter. So over time, this will cause these receptors called AMPA receptors that have been stored inside the cell and are not doing anything. It's going to cause them to translocate to the cell surface. And once the AMPA receptors go to the cell surface, that's going to allow them to bring sodium into the cell. And that's going to allow the cell to have a bigger EPSP in response to that same glutamate signal. So that's happening, showing this is happening here, right? So here we have the release of glutamate. Glutamate is going to bind to the receptor. We have sodium and calcium entering the cell. That's going to cause the AMPA receptor to translocate to the membrane. And now what's going to happen is that the glutamate is going to act not only on the NMDA receptor, but also on the AMPA receptor. So now when glutamate is released, the cell is going to have a bigger EPSP than it did before. So the more that this cell is stimulated by the original cell, the more sensitive it becomes to signals from that cell. And that strengthening of synaptic connections is going to be the basis molecularly of learning and memory.